Dear professors and colleagues, I'm sorry I can't come to the conference. Thanks for the chance to talk about our research on Selenium-regulated health management in cucurbit crops. I will organize my report around four principal aspects. First, here's the background. China leads the world in growing melons. It has 318,000 hectares of land, which is 37% of the global total. Each year, it makes over 30 million tons of melons. Xinjiang, Shandong, and Henan province together produce about half of that amount. Powdery mildew is a big problem for many crops like wheat, cucumber, grapes, strawberries, and tomatoes. In spring and autumn, more than 30% of crops get it. As a result, farmers lose 10 to 20% of their harvest. When plants get infected, they make substances like hydrogen peroxide and superoxygens. Then special enzymes like SOD, CAT, and POD break down these substances. At the same time, plants strengthen their cell wall with lignin and form callus patches. This acts as wall to stop selenium from spreading inside the plant. According to the most recent data from the China Pesticide Information Network, the National Pesticide Registry lists 9 approved active ingredients specially for melon powdery mildew management. There are 4 main types of registered substances: 1. Sexual fungicide, 2. SDHIs, 3. Mycin fungicide, and 4. A natural control agent, Bacillium subtilis. Each type works in a different way to fight the powdery mildew fungus. Each way to stop powdery mildew has pros and cons. Chemical pesticides usually work in just one way. The fungi can easily become resistant to them. Growing plants that resistant to the disease take a long time to breed. Mycin biosimilars with pesticides might help reduce the need for pesticides, but we still need to better understand how to use them and how they work. The following are the milestone events in the research and application of Selenium. In 1817, Swedish chemist Berzelius discovered selenium. In 1972, the crystal structure of the complex formed by phenylalanine acid was analyzed, thereby determining its three-dimensional structure. In 2020, the National Health Commission clarified that selenium can enhance human immunity. The primary mechanism through which selenium improves plant health is by starving the free radical generated by oxidative stress. Through literature-based network analysis, it was discovered that the application of selenium in methane plant health mainly pertains to bacterial or abacterial stress channels. Notably, selenium has been extensively utilized in addressing plant-bacterial stress. Studies show that selenium affects how plants make a certain substance. When selenium is added to soil, the stuff from rapeseed straw can stop cucurbit in rapids. It also makes levels of things like oxide and formic acid go up. For strawberries, nanosin can increase proline and boost the amounts of IAA and ABA. Plant secondary metabolites like cucurbit and polyamines directly engaged in plant defense mechanism. They play a critical role in enabling plants to resist pathogen infections. Further application of selenium fertilizer allows the selenium to enter the plant body via stormy stickers and other pathways. When compared to sodium nanoselenium with a single with puzzle size of less than 20 nanometers is more than effectively absorbed and utilized by plants. The second part of focus on the mechanism of nanoselenium induced resistance to powdery metal in melons. Firstly, nanoselenium was synthesized via chemical reduction method. Sodium silicate was gradually added dropwise to a Chatham solution under the action of the ascorbic acid. Chatham-coated nanoselenium was successfully prepared. The morph of the synthesized nanoselenium was observed using SEM, revealing that its particle size was less than 200 nanometers. Substantially, Team AFM and a particle size analyzer were employed to analyze the average particle size of the nanoselenium. The results show that the average particle size was 72.4 nanometers. Next, a portrait experiment was conducted. The leaves were sprayed with nanoselenium 3 times. 9 days after inoculating the powdery metal pathogen, it was observed that in the nanoselenium treatment group, the incidence rate and the number of disease spots per single leaf of 4 melon cultivars decreased. The control efficiency ranged 14% to 45%. In terms of the phenotypes of melon seedlings, the nanoselenium treatment group exhibit a certain increase in stem thickness and plant height. However, the treatment had no significant impact on the leaf area. Paraffin section stain was employed to observe the leaf tissue. After being infected by powdery metal, the sponge tissue within the leaves became loosely arranged, and the vascular bundles underwent atrophy. This change inflicted significant damage on the tissue structure of the leaves. Moreover, a substantial amount of mycelium was observable. Statistical analysis of the live tissue revealed that the nanosem treatment group demonstrated varying degrees of an increase in the thickness of the leaf epidermis plant-cell tissues, the ratio of plant cell to sponge tissue and tissue thickness. Sim and team were utilized to observe the leaves at different days post-disease. It was discovered that the number of mycelia on the leaf surface of the nanoselenium treatment group was pairs. Additionally, only the formation of mastoid was observed in the nanoselenium treatment group. Subsequently, the activities of beta-1,3 glucose and tryptase were measured. The findings show that the activity of GLU in the leaf of nanoselenium treat group increased at 2 different extents. The result was in line with that of the team observations. The accumulation of ROS was observed using a stem method. Results indicate that the ROS content increased following powdery metal infection. However, with nanoselenium intervention, the accumulation of superiorid, alline, and hydrogen peroxide were reduced. Moreover, the GSH, PON, and DPPH increased to varying degrees. Through the measurement of the activity of SOD, CAT, APX, and POD, along with the assessment of their relative gene expressions, it was discovered that the nanoselenium treatment could enhance the activity of these enzymes. This enhancement enabled the removal of the excess ROS, thereby improving the antioxidant defense capacity. Through ANSEC, it was found that differentially expressed genes in KGG pathway were mainly concentrated in secondary metabolite basins and plant organ single transduction. Assay mass was used to measure the IAA, GA, and SA. The results show that the contents of the IAA and SA increased to varying degrees after nano-Selenium treatment. Moreover, the signal transduction of the SA might be associated with the in vivo defense against pathway pathogens. In the field experiment, the combination of nano-Selenium and a thin recommended dose pyrazine along with the pyrazine demonstrate varying degrees of reduction in both the incidence rate and the number of the single lesions. The prevention efficiency ranged from 21% to 43%. When it came to cucumber, the control efficiency of this combine was 81% lower compared to that Bacillus subtilis as reported in the literature. This is mechanism of nano-Selenium intervention in melon tooth polymerial resistance. The third part prevents the mechanism of nano-Selenium induced resistance to cucumber green mold or studies in the library. Based on the literature reports, 5 different concentrations of genes were set. So pathotype observation and measurement of photosynthetic pigment contents. It was found that GSA treatment could enhance the biomass and increase the photosynthetic pigment content of cucumber plants. Research revealed that treatment with GSH enhanced the antioxidant capacity of cucumber leaves and facilitated the accumulation of phonatic acid. Subsequently, we select GSH and NanoC for combined application. Separate experiments were established, namely nano-GSH and nano-combined GSH. Through physician observation, calcifluence assessment, and determination of photosynthetic pigment content, it was evident that both the above ground biomass of total calcium content in the combined treatment group were significantly higher compared to those in other treatment groups. The activity of antihistamines associated with the cucurbit glucose cycle were measured. Research showed that in the group where nano-Selenium and glucose were combined, the activity of glucose peroxide and cucurbit glucose peroxide increased. This enhanced improved the agent activity capacity of cucumber and activate the ascorbate glucose cycle. The combined treatment of nano-Selenium and gelsin enhanced the synthesis of GA and cucurbit, while also upregulating the expression of their regulated genes. Subsequently, an inoculation experiment was conducted. After inoculation, the green mold pathogen, the diameter of the lesion were observed in measured research showed that combined with the control group, the combined treatment of nano-Selenium of glucose could significantly reduce the diameter of the lesions. After inoculation, turban blue stain and NBT stain demonstrate that following infection by the green mold fungus, the stain areas of the areas of rose stain was small. The accumulation of the hydrogen peroxide and MDA was lower compared with that of control group. Moreover, The combined treatment of nano-Selenium and glucose enhanced the activity of port. After bacterial infections, the level of second metabolites and omics were measured. It was discovered that the content of second mobilities, like autonic acid and quercetin, as well as Ga and Sa increased genes associated with synthetic photosynthesis and cucurbits in cucurbit were found to respond to the intervention of the actors G. Unsick indicates that the digits between the combined treatment group of nano and glucose, the control group, were predominantly concentrated in alpha-limic acid metabolism, immune biosynthesis, and twinkle metabolism. Post-inoculation, the digits were mainly enriched in plant pathogen intervention, peptide metabolism, and carbon metabolism. Metabolic analysis revealed that the DIMs between the nano and glucose combined treatment groups, the control group, were mainly concentrated in pathways such as alpha-lic acid metabolism, phenolide metabolism, and phenylpropanide biosynthesis, and amino acid biosynthesis. After inoculation, the DIMs were predominantly enriched in alpha-linolenic acid metabolism, phenylene metabolism, and amino acid metabolism. This is the mechanism of nano-Selenium and GSH intervention in cucumber to green mold resistance. I'd like to express the Xinjiang Academy of Agricultural Science. Thanks for your attention.